This method can help to demonstrate the differences between circulating T-follicular helper cell subsets in healthy and diseased states. The main advantages of this technique are that it's fast, that the samples can be batch processed, and that the cells remain alive for downstream functional analysis.
This technique facilitates the evaluation of circulating T-follicular helper subsets as biomarkers for disease progression, as they correlate with markers of clinical severity.
Helping with the demonstration of this procedure will be Dr. Lucia Piñon.
Begin by mixing 2 to 15 milliliter peripheral whole blood samples obtained in potassium EDTA vacutainer tubes with 50 microliters of a commercially available human CD4 positive T-cell enrichment antibody cocktail per milliliter of blood in one 50 milliliter conical tube per 5 to 15 milliliters of blood. After 20 minutes at room temperature, dilute the blood with an equal volume of 2% fetal bovine serum in PBS and slowly layer the diluted blood sample over 15 milliliters of freshly prepared density gradient medium without disturbing the interface. Separate the cells by density gradient centrifugation and use a Pasteur pipette to collect the enriched CD4 positive T-cell layer from the serum density gradient medium interface.
For a successful CD4 T-cell isolation, it's essential to layer the blood carefully without disturbing the interface and to quickly place the cells into the centrifuge for their separation under the appropriate centrifuge settings.
Then wash the cells 2 times in up to 10 milliliters of fresh 2% FBS and PBS per wash and resuspend the pellet in fresh 2% FBS.